we are so focused on what we see above ground, right? And that the only thing that matters is light. And this is such a frustration when you know that trees aren't usually limited by light. They're limited by nutrients or water, and those come from the soil. So we shouldn't be thinking about the above ground portion all the time. So once we started realizing this stuff about the importance of living roots, I thought, this is the message we've got to get across. There's that above ground half of the ecosystem, and there's this one too. And I think a lot of the benefits of retention forestry or continuous cover forestry are because they're living roots in the soil. So yes, there's cover, and that provides lots of nice stuff and has huge biodiversity implications. But below ground is at least as important.
Hey folks, what's happening? Welcome to Your Forest. My name is Matthew Christoph, and on this podcast, we talk about the environment and the science of sustainability. Now today, we're talking all about soil life, the life that is beneath our feet. Really cool. I've never talked about any of this kind of stuff, so it's new ground for me. Yeah, it was really, really enjoyable though. I learned a lot. So it turns out that soil is not just a substrate by which we hold up plants and hold nutrients that are decomposing. It's actually the other half of the ecosystem where so much life and interaction is going on. I'm talking way too much life, like more. You'll find out if you there's a lot going on down there. And it's only kind of just recently that we've learned just how much and just how active the interaction with the above ground ecosystem is. So we brought on two people, Cindy Prescott and Sue Grayston, to talk about the life in the soil and how it's interacting with trees and plants and that kind of thing, and how we can maybe use that knowledge to help create healthy soil microbiomes and maybe even sequester more carbon during forest harvest. So the two people we brought on, Cindy Prescott is a professor of forest ecology at the Department of Forest and Conservation Sciences at University of British Columbia. Her research digs into nutrient cycling and organic matter processes in forests. This includes nutrient availability and carbon sequestration, stuff like that. Yeah, really exciting. Sue Grayston, she is the other person coming on today. She is a professor of soil microbial ecology at the Department of Forest and Conservation Sciences at the University of British Columbia. Her research digs into microbial diversity and function in soils and plant microbiome interactions. So together, these two women have done a whole bunch of research, and they've discovered some pretty incredible things, some novel things to this world about how plants interact with the soil microbiome. And it's pretty incredible. It definitely changes the way I perceive the forest, right? Like I said, turns out that below ground is the other half of the ecosystem that we need to be considering. So we're talking about having sustainable forests forever, and that includes paying attention to what's going on below ground, the stuff we can't see. And the big thing that Cindy and Sue have discovered is that doing forest harvest, we need to be leaving trees on the landscape, about 30% on the landscape, to maintain healthy soil microbiome. So they're here to talk about that, to explain that whole concept and why it's important, what is going on, how we can maybe sequester more carbon in the age of climate change by caring about soil microbiomes. Lots of cool stuff. You're going to learn a bunch. Thank you to Cindy and Sue for coming on. This was really eye-opening. Really, really cool. Sponsors for 2024. Wes Fraser is the number one. They've been with me almost since the beginning. Yeah, I couldn't have done this without them. They've made this possible. So thank you, Wes Fraser, for your support over the years and continued support. I can't say enough. Thank you, Wes Fraser. And Greenlink Forestry. Greenlink Forestry has been with me since the beginning. And again, couldn't have done it without them. Thank you, Greenlink, for your support. All right, let's dive into the world of soil microbiomes. What is going on? What is the life beneath our feet? How much is there? What does it do? Why is it so mysterious? Here we go. Thank you for joining me early and late in the day, depending on where you are in the world. But I think we want to start off getting to understand. So we're gonna talk today about below ground ecosystems, about the life that lives under our feet. And I wanted to get both of you to just describe personally why you have dedicated your life to this below ground ecosystem, to this life that is there. So, Sue, just because you're on my left, and that's how we do it in the Western world, to start on the left, you can go ahead and start. Why is below ground ecosystems your life?
Okay, thanks, Matt. Well, I guess it started off when I was young, growing up in England, and my mum was a keen gardener, and I used to spend a lot of time outside with her, and started to get fascinated. She used to let me play in the soil, and I was really interested in the insects and things I could see crawling around in the soil. And when I went to college and university, I did microbiology, and I became fascinated once I could actually see those organisms under the microscope, things that you can't really see with the naked eye. And I was lucky enough to do a final year undergraduate project on soils, and had a really enthusiastic professor, and things just grew from there. I just became fascinated by all this living ecosystem with lots and lots of fascinating organisms that, as I say, you just can't see if you're just looking at the soil.
Yeah, a hidden world, right?
Yeah.
Okay, Cindy, for you.
Yeah, mine was quite different. I can't remember anything formative when I was a child except my love of forests. I just loved being in forests and would always be hiking and canoeing and spending all my free time in forests. And then when I finished my undergrad in biology, I got a chance to go to Alberta and work in forests, do research in forests, which I thought was just the greatest gig ever. So I just prolonged that as long as I could. And here I am, rather a number of years later, still doing it. But I didn't know anything about most of the functioning of forests. And so my master's project introduced me to the process of litter decomposition, which I'd really never, if I'd heard of it, I never thought about it.
Right.
And then I found out how important it was, and that was sort of the gateway to the rest of the below ground ecosystem. So I focused on the processes, and then became aware, of course, that it's organisms doing all these processes, but at that point it wasn't really possible to link the two. So people in the same lab as I was in were studying a lot about the organisms, and I and a couple others were focusing on processes, but we weren't able to put it together. But I always kept an eye out for when it looked like that might be possible. And it was actually some of Sue's work when she was in Scotland at the time that started using these new techniques. They were new at the time. They're quite old now. That gave us more of a functional indication of what organisms were there, especially microbes, and what they were actually doing.
Okay.
So I had the opportunity then to encourage Sue to come to UBC, and we formed the Below Ground Ecosystem Group. And our purpose there was to try to use all the new techniques to link all these below ground processes that are so important with the organisms that are doing that. So that's what we've been doing for the last 20 years through quite a number of students now who as a result are knowledgeable both about the processes and the organisms that are doing them.
Gotcha. So was it kind of a technological holdup? Like you kind of needed to wait for the technology to be available to understand what was going on? Is that what you mean by techniques?
Yeah, so I should mention when I was doing my PhD, all we could do was use culturing, so try to culture organisms onto agar plates, and we now know that only accounts for 1 to 10% of the total community that's there.
Okay.
So we didn't know much, and I always like to tell the students now, my first postdoc that worked with me in Scotland, when molecular biology started to come on stream, which is now enabling us to look at all the organisms in soil. Poor Chris took him 9 months to be able to extract DNA from these highly organic soils. And I was saying now to the students, you can buy a kit from Biorad and do that in 4 hours. So the leaps and bounds that have come on in the last 20 years have really enabled us to start looking at these organisms and what they're doing.
I would see all the other students in the lab with their entire lab bench was covered in these agar plates, and they'd just work endlessly. And in the end of the day, they'd come up with a list of the species or types that they thought were there. And I'd say, and what do they do? And they say, I don't know. So I'd say, okay, well, carry on.
Yeah. At least they know. At least they know they don't know. That's a big step in and of itself, right?
But as Sue said, even then, they, after all those years, they would just get a list of species that were there, which were probably less than 10% of what was actually in that soil. We now know.
Yeah. Interesting.
We now know they're the fast-growing organisms. So the slow-growing and the more rare species we know less about because, we were growing them under ideal conditions or giving them lots of carbon, which they don't see in the soil.
And you actually found a whole kingdom that was down there that we didn't even know were there, which actually explained a lot of what we were finding in processes. The soil has got a lot of these archaea, which we thought were just in hot springs and stuff, so we ignored them. And then we found out, once the techniques were there, we found out the soil was full of them.
Oh, okay. An archaea is a certain type of organism, species?
Yeah, so they were first thought to be some kind of weird bacteria, but once we had the DNA sequencing, their DNA sequences are too different from bacteria, so they're a separate kingdom on the tree of life. But we've now found out that some of them have a central role in, for example, nitrification, which is the oxidation of ammonium to nitrate. And like years ago, we thought there was just specific groups of bacteria that could carry out that process, but now we've found archaea can do that.
Oh, okay.
So we're having to rethink a lot of our nutrient cycling cycles. They're becoming a lot more complicated.
So it really is a hidden world, right? Like it's something that it seems like we've only just got in the last couple of decades to a place where we can really actually explore what's going on under our feet. What is going on down there? How it interacts? What's the ecosystem like? Do you think that does that account for why soil biome health wasn't really something that was largely taken care of, or it wasn't noticed really at the land management scale when we talk about forest management and that kind of stuff? Do you think that was why? Just because we just kind of didn't know enough to know to even start to measure it?
Yeah, I think if, as I say, it's kind of out of sight, out of mind. And then the problem is if you think about when we're looking at forest ecology, we can easily, identify a tree species by morphology, but you can't do that with microbes. So it's been very easy to ignore, I would say, because they're not, and these organisms, they're not cute and cuddly in terms of, if people think about above ground mammals, you can't ignore them.
We knew they were important for the ecosystem, but, how do you argue that when you can't really explain who's there and what they're doing specifically, just that they're important. But we don't know much about them. It makes it very hard to say we should consider this in our forest management when then, like the second question that comes your way, you can't really answer yet.
Yeah. We need that something to point to, right? Some kind of evidence to make the point in people's mind, put an exclamation point like, yes, it is important because of this, and we can measure it this way. So this is why, totally. And that's kind of why we're here today, right? Because I feel like both the talks that you guys gave that I listened to, they made really good points about how we can make that point about how, look, and this is why the soil microbiome is important. And this is how we know it's important, and this is why it needs to be considered. And I feel like this is it's a great opportunity to make that known. So
it's 20 years on now. The amount that we've learned and the rate at which our ability to learn about it is coming on. I think it's time now. Yes, totally. And you know what? I think there was one image in one of the talks. I forget who gave it, but it was a tardigrade. The water bears. I think that is the one fairly cute. I know there's a whole internet push to like the water bears, I think, right? So I think people would consider them cute, but I know what you mean about the rest of it. Okay. So I think we should get into, I would like to paint a picture in people's minds. I know nothing about, below ground ecosystems. I imagine a lot of my listeners are probably the same way. I would like to paint a picture of what is going on below ground. What kind of life exists there? Yeah, what is it made of? What are we talking about?
I would suggest looking at it through the lens of what they're feeding on and dividing it into two major groups. That is the ones that we studied for longer, which is the ones that degrade or plant organic matter.
Okay.
So any plant litter, be it the leaves, the woody material, the roots, there's a whole bunch of organisms that use that as their resource.
Okay.
And as a result, when they break that down, they release all the nutrients in it eventually, which the plants can then take up. Otherwise, the plants would become starved of nutrients. So there's, shall we call them the litter degraders.
Okay.
And then the ones that are associated with living tree roots. So I now see them as two equally important but distinct communities. So there's a lot of organisms that live on the surfaces of roots or root tips, as well as the mycorrhizal fungi attached to the roots, and all the organisms that live either on or on the surface of these mycorrhizal fungi. And each of those sets of organisms sets up a whole food chain.
Ah, okay.
So in both of those systems, there's a whole complex web of organisms. Like we see above ground with food web, we're very familiar with above ground. There's at least two very complex food webs below ground.
Gotcha. So that's interesting to point out. So because I think myself, I would have thought of soil microbiomes, like whatever's living in the soil below ground, to be mostly related to decomposition. But there is actually interaction with living organisms that have above ground biomass. And so it's not just that it's decomposing, but it's actually providing active feedback somehow or another. We'll get into that, back to the living or plants, organisms as well, which is cool. I think that's another layer that I think most people might like, unless you're, in the field that you're in, probably wouldn't know, right? So very, very cool.
Yeah, it's not well understood. And so through the let's say the 70s, 80s, 90s, the focus was almost exclusively on the litter decomposers, and that's what I also focused on. And then there came to be compelling evidence from some of the carbon tracing studies that showed that up to half of the carbon that's fixed, the CO2 that's fixed in the leaves of the trees, it is sent below ground.
Half.
And it very quickly, within hours or days in the case of trees, comes out of the soil as CO2. And it's not all being just respired by the roots. It's also being taken into the mycorrhizal fungi and respired by them. But also now we know going out into this food web, which we really didn't understand until recently, and being used as the energy source for all those organisms that are living on this sugary labile material and releasing that after they metabolize it as CO2. So half of this CO2 that's coming up from the soil was very recently, within days, up in the leaves of the trees. And, a day before that was up in the atmosphere as CO2. So very dynamic food web.
Dynamic and fast moving. Yeah, for sure. That's interesting.
And huge. Because another thing is if you think about the numbers of these organisms. So if you were to take a tablespoon of soil, you've got billions of bacteria in there. So there's more organisms than there are people on the earth in that tablespoon. So
isn't that ridiculous?
Billions of bacteria and a couple of 100,000 fungi and maybe 200 nematodes. And so thinking about all the organisms, the larger ones are smaller in number, but they're feeding on these just masses of trillions of bacteria that are down there in the soil.
That is wild to think about. Like a tablespoon of soil contains more microorganisms than there are people on the planet. You're like, well, how do you, the human brain is not capable of, it makes you, okay, so if there's that much in a tablespoon that you grabbed from wherever your research study was, and there's how much soil on the planet that there's no chance of us studying all of it, how much are we missing? Now all of a sudden the idea that 1 to 10% of the soil microbiome, who knows? It makes it, I see what you're saying. That's crazy.
We can now with the next generation sequencing techniques, we can look at sequences of every organism.
Oh, like genetic sequences.
Soil.
Okay.
But what are they all doing? We still don't know that. We can do, from those sequences now, we can do things like predictive metagenome profiling. So we can search the databases and look for functional potential in there. So that would give us an idea of what these organisms can be doing. But we're having to deal with a lot of big data now. So it's making sense of these, millions of species in these billions of organisms. So yes, it's a huge job to actually try to understand.
Yeah, no kidding, right? That's just those numbers
unimaginable
alone.
Only 20 years ago that we'd have our hands on these data.
Yes, no kidding. Technology is amazing, right? Who knows where we'll be in another 20 years? How much faster we can go through that data or whatever it might be, right? That's very, very cool. So, is it fair to say? We've only recently kind of really started to map out what's going on down there? Is that a reasonable thing to say? This is relatively new science, and this is something that's not well known across even environmental sciences.
Yes, more and more is becoming known, and what Cindy was talking about with we're now realizing how important the labile carbon is from plants. And that's really been through some of the stable isotope, the labeling techniques that we've been able to do again in the last few years. So we can label trees with heavy isotopes of carbon sources similar to the carbon that's coming from the tree. So glucose or amino acids, and then follow that into the microbes. And in that way, we've been able to identify organisms, groups of organisms that are really important in using that carbon, and then follow them being eaten by some of the soil fauna.
Gotcha.
And,
those studies really changed the picture, because we realized, wow, look at this stuff moving. It's just flowing from those leaves into the roots so quickly, and then out into the mycorrhizal fungi, which we expected. But then Sue's work and other work has shown it starts showing up in all of these organisms in the soil, which I had to say I didn't expect. I thought, they share it with the mycorrhizal fungi, end of story. And that's sort of what we all thought. But then we found out that they're actually exuding it or being grazed or whatever, and that carbon is moving quickly into the ecosystem. And if we do another number we have now is thinking about how much of that soil is actually in the zone where you would have the microbes would have access to either roots or mycorrhizal fungal hyphae. On average, in a cubic centimeter of soil, so imagine that is sort of like your fingertip, where your fingernail and tip. On average, there's 7 centimeters of root in there, mostly fine roots. But there's 2,000 centimeters of fungal hyphae.
That doesn't even make sense.
You can't imagine how there'd be room for it. But then when I saw numbers like that, I thought, and if they're exuding carbon and sugars out of them, and who knows what all they're exuding, and we know that there's bacteria in that lining the hyphae, then this is a major flux, potentially a major flux of carbon out into that food web directly from the tree roots and mycorrhizal fungal hyphae.
Yes. Yeah. These are concepts that it's hard to even like wrap your mind around, that in something as small as a cubic centimeter, there's 2,000 centimeters of, you said fungal hyphae? Like it's what? Like I don't.
Yeah.
It paints an interesting picture. That's for sure, right? Like it's just wild.
It's hard to conceive of.
Yeah, absolutely. But I'm glad you're here to help us conceive of it. So
the other problem is that it is so hard to, most of it you can't see.
Right.
So I've been really enjoying seeing some scanning electron micrographs that they get of tree roots and fungal hyphae, and you can see the bacteria on them, because it's so important for us to be able to appreciate it, to be able to visualize it. And so that's always been the problem in getting people interested and concerned perhaps about life below ground, is you can't see most of it.
Yeah, so who cares?
The really important ones are almost impossible to see. Not anymore, but you can at least get little snippets of where they are.
Yeah, absolutely. Well, going off of what you just said, the why should I care thing, I often start with that when I do these podcasts, is like, why should people care about these below ground ecosystems? And the next step that we're going to go to is going to be talking about how disturbance, so whether it be forest harvest or fire or whatever it might be, affects these ecosystems. But before we do that, so I want to talk about what these ecosystems are doing for the things that we can see, that humans are so good at caring about, like making sure that trees are coming back, making sure that the big megafauna, the cute, cuddly animals and insects that are important, like bees and stuff like that, that people seem to just, climb all over their importance and set up a cause for them, get a charity going. I want to know, how do these below ground ecosystems affect the things that people already do value and care about and think about? So if we can talk about that in some way, I'm not sure how you want to do it, like maybe how trees might be affected, or, above ground health, forest health might be affected by a poor soil microbiome or whatever it might be. I'm not sure how you want to put that into context, but why should people care?
Yeah, maybe I'll start with an example that most of us know, that birds like to eat grubs. They get grubs and worms out of the soil.
Yep.
But what we don't know is how many insects actually spend part of their life, a good part of their life, below ground. And so there's soil fauna that always live below ground, and there are also soil fauna that spend part of their life below ground. And so just to form a link with above ground biodiversity, there's all of these insects that spend their larval stage, or whatever stages, below ground.
Beginning stages, yeah.
and the birds love these, and probably other organisms as well.
Right. Yes. It's there must be so much interaction too with like you were talking a lot about like carbon cycling and nutrient cycling, and like at the very beginning someone mentioned that if it wasn't for, or Cindy, I believe it was you mentioned that if those decomposition organisms didn't exist, then the plants would starve, right? They would die out.
Yeah, so and that was a very important evolutionary step, was for the fungi to be able to have the enzymes that enable them to break down the more difficult to degrade part of plant cells.
Right.
So if it wasn't for that, all of the plant litter would have just built up and accumulate, and if there isn't that release of nutrients, then it would have been over, because the trees are depending so much on the nutrients that are released from their own senescent and dead tissues.
Yep.
That if those microorganisms weren't able to do that, that would have been the end of the development of terrestrial ecosystems.
Right.
Yeah, we'd be buried knee deep in litter.
Yeah.
Yeah.
So they're really important recyclers of nutrients, but they also produce and consume gases that are important for global climate. So we have certain microbes that produce methane in waterlogged areas, but our forest soils are important methane sinks.
Okay.
So they actually remove methane, which is a more potent greenhouse gas than CO2, and oxidize it. So removing that from the atmosphere. There are also denitrifying organisms that can remove nitrous oxide, convert it back to nitrogen gas.
Okay.
The basis of our waste treatment is microbes.
Oh, okay.
So we'd be buried in waste and feces if we didn't have microbes. And thinking about these organisms that we haven't cultured yet, and the ones we're still to discover. Our first antibiotics came from soil organisms. So Penicillium notatum produced penicillin, and you have streptomycin from streptomycete organisms. And there's a lot of, interest now. We shouldn't be using pesticides, herbicides.
Okay.
And there are potential biocontrol organisms in the below ground community. So, people are looking at green chemistry alternatives. So using microbes and soil fauna to control pests.
As a replacement to pesticides. Gotcha.
Yeah. So, using some nematodes to control so nematode worms controlling insect pathogens.
Okay.
So if we could understand what all these organisms do, then we could use that knowledge rather than chemical control to have biological controls.
Think about the untapped
soil
potential,
fauna,
right?
Another thing they do is very important because they can break up the litter. They don't just eat the whole leaf or needle. They eat certain parts of it.
I see.
But that creates the access for the microbes, the bacteria and the fungi that can then get in there and do the decomposing further through their enzymes, which do the chemical breakdown eventually into nutrients. And another thing they do, which I found out, I guess you'd say the hard way, is these little soil animals, they release the nitrogen in the soil. Yeah. And I found that out because I was doing a long-term microcosm study with some forest floor material, just humans that you collect from the forest, just in the lab. And then I was extracting them periodically to see how much nitrogen was being mineralized. And these pine ones, I found, never released any nitrogen. They lost half their mass, all these needles and such, lost half their mass and never released nitrogen. But what I noticed is the pots became just full of fungus.
Oh, yeah.
And so I realized, unless you have the grazers there, the fungus is just going to go ahead, do its business, just hold on to all that nitrogen. And that's when I realized, if these aren't grazed, then it all just sort of stops there. They just get fat and happy and just slowly senesce. So that's another service that a lot of soil animals do, is just to get that nitrogen moving again.
Gotcha. Yeah, so you need the whole system. You can't just select a few microorganisms to do the job. You need all of it to make
especially when you realize that these collembola and such, these tiny I guess they're visible to
springtails.
Springtails and mites. They're quite particular eaters, and they tend to specialize on certain fungi. So if you got a whole lot of species of fungi, you also need a whole lot of species of these megafauna to graze on them to keep that nitrogen recycling, because that's what's limiting tree growth. So
you need fungal predators.
All these organisms doing their thing, and then you get nutrients being recycled, and you get trees able to continue growing.
Yeah. Okay, gotcha. Yeah, that's really, really cool. It's good to paint that picture, right, of the importance of the whole system as one piece, and not separating it and thinking, if we have just this one organism or whatever, we're good. But no, you need the whole system. And to the earlier point, regarding what Sue had mentioned, think about the untapped potential of solutions out there to who knows what, right? That's in the soil, that comes from these microbes, that we don't know about yet, that we haven't found yet. So there's, and we're starting to map that out. Like this is everything. It's really, really cool.
And for carbon sequestration, I mean, the microbes, the soil organisms are so important for that, because I think it's about half of the carbon in forest ecosystems is sequestered in the soil. And it's constantly being degraded and released as CO2, which means it has to be continuously replenished. And we thought this would happen, through litter, and it does. But we also now know that there's the other half of the story is this microbial processing. They're essentially dead bodies or dead cell envelopes, and these weird compounds that they release. These are sort of the building blocks of soil organic matter. And because they're right down there in the soil, and that's what creates the most persistent soil organic matter. So it seems funny, but in order to get this persistent organic matter being continuously replenished, you need to have this labile carbon coming down, feeding the bacteria, and a portion of that becoming new, persistent organic matter that gets complexed with the soil minerals and lasts a long time. So we need to think not only about how to prevent carbon from being
mineralized, or organic matter being mineralized, and so the carbon released is CO2, but how to keep that constant flux coming in. And that's why I think we need to think more about the role of living trees in creating soil organic matter and sequestering carbon in the soil. So in order to sequester carbon in the soil, you've got to understand the whole process by which it's both created and then degraded. And for that, you got to understand these, especially the soil microbes, I think.
Yes, of course. That makes sense. So, carbon is something that unfortunately, I still haven't had too many really robust conversations about forest carbon. And so let's talk about that relationship between the forest carbon and the below ground ecosystems a little bit more.
Yeah. So I'd say 20 years ago, at least before 2010, which is when I started noticing what was being found by the soil organic matter chemists, we thought that the main source of soil organic matter was litter, right?
Right.
Dead plant tissue, either leaves, needles, roots, or wood. And so the focus was on how to make that more difficult to or slowly decomposing, and so
to
that
not release
was
the carbon.
The focus. Can we increase its lignin content or reduce its nitrogen content or do something that'll make it more slowly decomposing? And that should, at least for a while, result in there being more organic matter and carbon in the soil, because it's all 50% carbon. So any plant litter is about 50% carbon. So just get more litter and have it decompose more slowly. But then the soil organic matter chemists were finding that most of this, especially the more persistent organic matter, it doesn't have a plant signal anymore. So
right, because you can't see it. You're going off of the like radiation signals or whatever.
Nuclear magnetic resonance spectroscopy.
We all understand those words. Those make sense to me. It's fine. Continue talking.
I can't explain it myself, but all I know is what they found. The people that understand that, and they found, hey, this isn't plant stuff anymore. This has got a microbial signal. It's being processed. So rather than the microbes kind of being the bad guys in that they're breaking down all this good organic matter that we're trying to keep there, they're actually the good guys. They are processing it and actually making part of that more stable. And that's what we're showing up in the soil organic matter in the persistent pools. Which has become associated with the individual particles. So it's quite small, and it's really deeply immersed into the soil substrate. This stuff is already processed. So what we need to do is not think about decomposing, having stuff decompose more slowly, but actually get it processed and have a greater proportion of it converted into material that then becomes persistent.
Right.
So that was a real
so it's
game
locked
changer.
In. So that those microorganisms actually lock in the carbon into the soil for a longer period of time than if it was just
Yeah. I mean, depending on where it is and who gets there, other ones have the key to unlock it in their enzymes. But if you can get it either put into aggregates, which are created by the secretions from the roots and the fungi, and that kind of sticks stuff together. And so if you get these aggregates, then some of the material that's inside that, it's hard to get. And so that's a lot of the challenge with sequestering more carbon in soil is how to reduce its access. So you can get physical separation from the organisms that might be able to degrade it, or you could get chemical fixation where it's actually bonded to especially clay minerals. Either of those will make it less accessible to the microbes. So that's the other part of the story. So it's not just about having stuff decompose more slowly, because eventually that does decompose. You got to get it locked in somewhere.
So it seems like by promoting, and correct me if I'm wrong, but tell me if this is fair to say. So if we can maintain the biodiversity, we'll use the term biodiversity here, of the soil microbiome across changing forest landscapes, that will help lock more carbon into the soil?
Potentially.
True
Because
scientist.
This labile material will also be used by some of the microbes to give them the energy to generate the enzymes that they then use to break down organic
But you need that
matter.
biodiverse system to make that happen. Okay.
Yeah. So it really is really tricky to predict. But now that we know about this flux and how it generates all of this activity, some which sequesters carbon and some which releases carbon, now we can get to if and how we can manipulate that for whatever purposes we have, one of which might be carbon sequestration. Because if
Seems
we can
like an
get
important one.
it tucked into mineral-associated organic matter, that's the best chance for having it stick around longer. Because trees are here today, gone tomorrow. But if you can get it into that soil matrix, you have a very good chance. But we're not there yet.
Gotcha. So healthy soils gives us a better chance to, healthy soil microbiomes, I should say, healthy below ground ecosystems give us a better chance at a more stable carbon flux, I guess. So like, I think about it, I'm trying to think about it as like a carbon sequestration solution in the age of climate change, right? We're always talking about how do we sequester more carbon? And not that this is how we solve the big problem, but every little bit, it helps, right? So I'm thinking about this as a way of maybe how can, say, forest management, for example, harvest differently to try and support more carbon being sequestered into the forest soils, and maximize the amount of carbon sequestration, and also, maximizing the health of the forest ecosystems and all that kind of thing. So I guess we should talk about the resiliency of these microbiomes, okay, and how they relate to different disturbances. So I know in a lot of your studies, you were doing stuff regarding, in clear-cut forests and other types of harvest strategies. How is this soil microbiome affected by, let's say, forest harvest? Pick your version of forest harvest you would like to go through.
Okay, so, we've been able to show from studies that zone of influence of the trees is really disappearing by about 10 meters. So if you're considering what we're doing in a lot of BC is clear cutting, that the amount of carbon that's reaching the microbial community that's, as we've discussed, is very important.
Right.
For nutrient cycling, for carbon sequestration, you're losing that fuel for the microbial community.
The second portion, the living portion of the microbial community that's dependent on living root systems, right? Yes. Okay.
Yeah. So we've done studies looking at clear cuts, at aggregated retention, where they leave patches of trees, and dispersed retention, where there's single trees left on site, basically like thinning the stand.
Sure.
And with the aggregated patches, we were thinking at first they would retain the soil microbes and their functioning, because you have more trees there, more roots, but the dispersed retention has a much better function in terms of retaining the microbial diversity and its functioning across that cut block.
So that being the individual trees, like spaced out, kind of like more like a thinning idea. So like
Yes.
instead of clumps of trees, you've got, individual trees spaced out every 10 meters or whatever it might be. That was the better.
Yeah.
Okay.
And you've done some actual transect studies tracing that carbon out into the...
Yeah, so we did some studies going out from the center of aggregated patches of retained trees.
Okay.
Going out 30 meters into the clear-cut area.
Okay.
And we were able to show that the microorganisms in the soil were using carbon from the tree.
Okay.
But using 13C, we can now follow where the carbon that's been fixed by the tree, where it goes to in the soil. And so we've been able to do this to follow the signature of the depleted carbon from the tree into the microbial community. And so we were able to make these heat maps, basically showing where microbes in clear cuts get their carbon from.
Okay.
And going out sort of once you reach 6 or 7 meters, that the microbial community there from the stem of the tree is being fed by the carbon from the tree.
Okay.
But once you get further than that, the microbes don't contain that depleted 13C. So we knew how important the living trees were for supplying microbes.
And then I looked into the mycorrhizal fungal distribution with relative to living trees.
Right.
I just looked into it in the literature. Fortunately, a few people had done all the hard work to get the information. But they very consistently find by the time you get 5 meters from the tree, the amount of mycorrhizal fungus is really reduced. And by the time you're 10 meters from a tree stem, I should say, they're just not there anymore. So Sue's stuff kind of showed that this goes for the whole soil microbial community. So beyond 5 to 10 meters certainly beyond 10 meters from a tree stem, what you've got is the community that's living off the litter, which is mostly fungus, and the organisms associated with the saprotrophic fungus, the decomposer fungus. So you've got that community, but you don't have this whole community that's associated with living tree roots and living mycorrhizal fungi. Okay. So they are so dependent on this very dynamic flux that as soon as that tree above ground portion of the tree is gone, basically that whole flux is going to stop. So this information had caused me, when Sue's students were coming out with this, I don't know, in the last 5-10 years, maybe 5 years ago, now I look at ecosystems very differently, because when I'm walking in a forest, I can visualize this very dynamic flux that's going on all the time. And then when I walk out of the forest and into the clear cut, I think, whoa, how did that whole system below ground just change? And now with retention harvesting, often there's 1 or 2 tree stems. So you walk out into the opening and you look at that, and now I can visualize that. So that's what I encourage people to do now that we know about that, is just visualize this dynamic flux from the tree to its leaves to its roots, into the mycorrhizal fungi, out into the soil. You can see that with each living tree that's retained. And then the spaces between them, if they're more than 10 meters from a tree stem, there's none of that. You've got a lot of slash there. You've got some fungus degrading that, maybe some things chewing it up. But, you've got wood that's going to be there for a long time. Yeah. But you're into a very kind of slow cycle there compared to this very dynamic system you have with living trees.
That makes sense. I never thought about that. So even the the logging slash that's left on the landscape is going to be decomposing much slower in those in-between spaces where they're more than 10 meters from a living tree, living root systems, versus the ones that are closer are going to decompose faster and maybe contribute more to the flux, the carbon flux of life, as you so eloquently titled your talk.
The communication people picked that one. It's not something I was really comfortable with. But it's a good name, I think.
It's a good name. Sometimes you got to do that in media. You got to get people's attention somehow.
Yeah. So that's what I would encourage people to think about when they're next out in a forest, is just try to visualize this. I used to do this with nutrient cycling, with litter coming down and then it decomposing, and, imagining that that's going on below ground. Half of the biomass of the tree is below ground, and there's always fine roots senescing and dying and being decomposed. So you can kind of visualize all that happening, and then the organisms eating that material, and the eating those organisms that are eating that material, and so on. But then you add this dynamic flux to that, and you can just see there is so much going on in there. And then you add the other part, which is eventually these nutrients being released and the trees pulling that back up to the leaves. There's so much going on all the time at any moment. And so I encourage people to just try to visualize that all of that is not just green stuff with some stems. There is so much going on in that whole ecosystem, only half of which you can see. Yeah,
that is incredible. Absolutely. And so I would like to think about, in the discussion, what about, so we're talking about tree roots, living tree roots all the time. What about other plants? What about shrubs, forbs? Is their role similarly important, or are trees? It is. Okay.
It is. Every plant in there is important for this. They are all fixing carbon. They are all exuding carbon into the mycorrhizae and into the soil. Okay. And we also know that each species has their unique, different, particular mycorrhizal fungi that associate most with them. And, the diversity of mycorrhizal fungi just on one tree root or plant root is also mind numbing. So it's not like just, I'm your mycorrhizal fungus. There's, I don't know, hundreds of them. But anyway, there's all that going on, and that's going on for every plant in there. And they're also exuding different things, different kinds of compounds. And there's been some really interesting work. Well, Sue did some of the early work on this, actually capturing those exudates. And you can see that what is coming out differs among all the different species. And now we know that then sets up not only differences in the mycorrhizal fungi, but differences in the soil microbes that are associated with them. So what we found is that because of this, if you have an increase in diversity of plants, you have an increase in diversity of soil microbes. And then because that feeds through the food chain, then you have a whole increase in diversity of soil organisms in general. So yes, the trees are important, but it's also the diversity of plants in that ecosystem, especially if they're in different functional groups. So if they're different. So conifers are kind of more similar to each other than they are to broadleaves. Right. And those are more similar to each other than they are to plants that have nitrogen fixing microbes associated with them. They're a very different group in terms of what they exude. And then forbs and grasses, they're also exuding things. They'll have their own microflora, the shrubs as well. So that's another thing I try to visualize, is just if you have all of these different species, they're all doing this carbon flux thing, and they're all associated with somewhat different microbes and soil fauna below them. So if you add that all up, just in that patch of forest, you can just imagine this below ground diversity that you have. Mm-hmm. And, if you can have diversity above ground and below ground, you have to have a more resilient ecosystem.
Yeah, it seems like that's what we're headed for the end goal for forest management as far as sustainability has to be hedging our bets, right? Has to be resiliency, and what I'm getting from you is that biodiversity begets biodiversity. So that's hedging our bets a little better, and there's a lot of unknowns with all kinds of ecosystems, and this one as well, right? So
simple ecosystems are really super easy to manage, for timber, but they are so risky now because you just don't know what the next thing to hit a forest now is.
Especially in the age of climate change, right? When things are changing so quickly, and you never know what pathogen or bug is going to just explode the way, say pine beetle did all of a sudden, right?
Yeah, that was an indication that this is not the same game we were playing.
Yeah, things have changed.
In 1990, everything was fairly simple, and then the pine beetle hit, and then what is it now? Record wildfire. Well, look at Alberta last summer. Good grief!
Yeah,
who would have imagined?
It was a mess. Even Canada-wide, I think it was. I think we doubled the previous total wildfire burned area in Canada last year. Yeah,
and you think, that was bad, and then you think, what's next year going to bring?
Yeah,
and
exactly.
if not next year, then the year after that.
Yeah,
and then we've got
resiliency is key.
Yeah, atmospheric rivers. You got so much flooding as a result of clearly related to forest cover is the relationship with flooding. So all of these things and record temperatures. How are these? Every species of plant has its kind of zone of comfort with temperatures. So if you've only got one species in there,
you're in trouble. Yeah,
I think so.
I do want to. So you talked about the importance of biodiversity as far as above ground plants. Are trees somehow more robust? Or more I'm trying to point out, are trees more for example, say you were a forest manager or whatever, and you're trying to maintain soil ecosystem health, but you want to minimize the number of trees you leave in the landscape. Could you just leave? I'm not suggesting we do this. I'm just looking for the holes in the argument kind of thing, right? Could you just leave a bunch of shrubs, of diversity of shrubs, pull the trees off, and that would maintain? Or are the trees somehow more have more weight to them when it comes to keeping things healthy? I know this is a hard one to answer. I'm asking a lot, but I want to
vastness of their root systems and mycorrhizal systems, as well as the depth of them, because that's the other thing about soil. We talk about 5 meters of the stem, but we're talking volume.
Yeah,
anytime we're talking about soil. So also the depth of those, and the difference between a mature tree and a young tree is there's a quantitative aspect, but there's also the qualitative aspect, because there are certain fungi and such that are associated with old trees, which is different from those associated with young trees. So if you're also managing for a single age class, then you're reducing the diversity.
Right. So diversity of age class is also important. Yeah. Okay.
So we can do this by doing partial harvesting, and allowing other species to be in that space with them, especially the native ones that come in naturally. Let's give them some consideration. But you can do it on a landscape level as well, just to keep at least the inocula of those species associated with older forests in the vicinity, so that they can come back in as the new forests mature. And this is not only for our logged sites, but also for all these wildfire disturbed sites. We need to, it's kind of quasi-natural.
Sure.
The wildfire.
With an asterisk, because of humans. Yeah,
what's wild anymore?
Yeah. Sure.
But we should be thinking about those
as well, and what we want to bring those back to, and can we make them more resilient by making those ecosystems more diverse, rather than just how fast can we get some trees growing on this site and make it look green. How can we build a resilient ecosystem on this site?
Yes,
well, we solve the other problems, like maybe not having record wildfires every year.
Yeah. That's a bigger problem that requires a few more solutions, I think. Yeah.
And we don't have them.
No, I'm not asking. Yeah. So you did stumble on the next thing. I would like to compare disturbance types a little bit. I'm not sure how much research has been done into disturbance types and how the soil microbiome differs based off disturbance type. So we talked about kaplaks, I think quite a fair amount. What about natural disturbance? Obviously, fire is the big one for Canada and for places like Australia and the States and stuff like that. So how does that compare? If you were to compare the two, like say a clear cut versus a big clear cut versus a big hot burn, how do they compare?
Yeah, the hotter the burn, the more devastating it is for the soil biota. Maybe Sue, you know more specifics than I do, but we know there's quite a reduction, in the microbes that comes with all the changes that happen after a severe wildfire.
Yeah. Sue, can you shed more light on that?
Yes. So if you have a really severe, intense wildfire that burns really hot, takes off all the forest floor, then you can be losing all those microbes that reside in the forest floor itself.
Okay. So that is a natural process, natural being quotations, but yeah, okay.
Yeah.
Okay.
Yeah, and it's fine on a landscape scale to have some of those areas. It's just that if they get to be so much of the landscape, which they're threatening or are doing now, this is when we have to be concerned about it. Because landscape diversity is good, so having some natural disturbances of various intensities is good. Mm-hmm. But this is something we could never have imagined.
Yeah, it's a different scale. Yeah,
and then if you have areas, as you say, across the landscape where you have unaffected forests, they can act as inoculum.
Okay.
To
or seed zones,
recolonize.
Yeah.
Those areas.
Okay.
And there's some neat work done at University of Alberta with up in the oil sands, which is inoculating these recreated soil substrates.
Yes,
with forest floor from surrounding forests, and just kind of helping get that back. And eventually, the spores would get there, probably, but it's slow and kind of happenstance. So you can actually inoculate. So maybe in these severe wildfires, it depends, because it also changes the physical and chemical nature of the soil, the upper soil. Mm-hmm. So it might help.
Yeah.
And but what's going to really help is getting the trees and other plants back in there, maybe especially nitrogen fixing species, because really hot fires just blast off. In all that forest floor is a huge component of the ecosystem's nitrogen. Mm-hmm. So they're going to need to have some nitrogen put in there. So getting plants in, and especially getting plants with nitrogen fixing associates, I think we should be focusing on rehabilitating these soils like we usually do with mine soils and things like that.
Yeah.
Because they are, even if it's quasi-natural, it's going to take a while to rebuild that site, especially if you want to have a fully functional and productive forest on it. And if you want that to happen sooner rather than later, I think we better start thinking about what in addition to trees we want to put in there.
Absolutely. That makes sense. That totally makes sense. I kind of want to I have a few more questions along this line. I kind of want to set a baseline of understanding for So let's take pick a disturbance. Let's go with fire, right? Let's say a bad fire. How long would it take for that soil microbiome to not get back to what it was? Because I realize, if it's an old forest, obviously it's going to take the age of it growing back to that old forest to become the same. But let's say, how long does it take after a fire for that to come back on its own sans people? Just to create some kind of understanding.
We know about after harvesting, and you're talking.
Oh, sure. Do that then. Let's do
30
that.
30 years or
okay.
so. And
why 30 years? Why that?
It's just what the evidence has shown, and that's kind of related to soil carbon levels.
Okay.
And we know from the work in mine sites, both in Alberta and Czech Republic, that after about 30 years, especially if you get nitrogen-fixing species in there, you're starting to get the soil organic matter building up again. That's the other thing nitrogen fixers do, is increase the rate at which soil organic matter gets back in there. And then if you also assist with maybe inoculation of that forest floor as it's developing, so that you get that functioning. In those situations, most of the organisms required for functioning soil are there within about 3 decades, and that's in a temperate ecosystem. In a boreal, probably longer than that. After severe wildfire, I don't know if we know that because of all of the other changes to that.
Yeah, like if you nuke the organic material and it's just mineral soil, like how long does that? Like it's got to be decades at least, right? Like,
and then if the fires are repeated, like some work Suzanne Visser did in the northern Alberta jack pine woodland ecosystems, jack pine lichen ecosystems there, because it's, I guess, very dry and it burns hot. You basically start with just mineral soil, and then that slowly starts to build, and then burns again, and slowly starts to build, burns again. And in those systems, you have such low availability of nitrogen that it kind of leads to this probably progressive decline in productivity, because the system cannot hold nitrogen. It gets up in the trees, it gets recycled, that's burned. And they start over again. So
almost like a natural deforestation in a way, where over generations it could become like a shrubland or something. Yeah.
So it creates its own ecosystem, those sorts of conditions, which is fine, again, on a landscape scale, on a natural scale. But now if we're getting into systems that are going to be repeatedly burned, I really think we need to think a lot about nitrogen fixers and getting them to help us rebuild that soil organic matter and nitrogen, so that the site does not decline. If there are repeated wildfires that are hot and are beyond what that system naturally had, it's going to change. It's going to become a poorer ecosystem.
Give me some examples for folks of some nitrogen fixing species that are plentiful in the boreal or temperate forests.
Yeah, any of the alders. So on the coast we have an actual tree, red alder, which is dynamite for putting nitrogen and organic matter into soil. Does it very quickly. But certainly in Alberta, there's the green alder. I think the Sitka alder.
Green and I think green and river alder. Yeah.
And Schipperdia.
Oh yeah. Buffaloberry.
Yeah. That's a nitrogen fixer. So so you would have to rely on shrubs and probably lupins, things like that.
Okay. So it's very important for shrubs to exist, not just trees. Like we definitely need those as well, for sure.
Certainly after wildfires, to get the nitrogen back, hot wildfires, if the forest floor has been burned off, then I think you would want to get nitrogen fixers in there to build up the soil organic matter and the soil nitrogen to keep that system productive. Otherwise, the trees are going to be less productive as nitrogen
declines.
Interesting. Yeah. And my point in bringing up the fires and comparing the two to see is not an attempt to try to say, hey, well, if fires do this, it's okay for forestry to do that, right? I think it's kind of the opposite. Like I feel very strongly that if we're capable of not destroying that soil microbiome by simply leaving some trees and shrubs around, like why wouldn't you give, like, a lot of times you want to give the forest a head start. Why not help it along? If something as simple as retention harvesting can help us in that way. Like I think there's a lot of discussion right now about the concept of emulating natural disturbance, because that was what we said we were doing for a long time. And I think we're kind of moving away from that. We're recognizing that humans,
we need to move away from that.
Yeah, we're not natural disturbance.
That made total sense in the 90s.
Yeah.
When we're also focused on sustainability, and we could imagine some sort of constancy.
We're hedging our bets somehow. Yeah.
That's over. It's so over. We need to think about these ecosystems and how we can help them. Yeah.
Yeah, forest harvest is so much more intentional than any kind of natural disturbance that we need to have intentional, also not just intentional, but it's so much more intentional and so much different than a fire is, right? Or a wind blow or whatever it might be. So I think that because of those two things, we really need to be spending the time to actively promote forest health and not just trees. And I think that
we need to think about
most foresters will agree with that.
Yeah, restoring and rehabilitating, regardless of the disturbance. Mm-hmm. We now should be able to figure out how we can make this a more resilient and possibly productive ecosystem, but primary goal should be resilience. And if that's living with the fact that there's going to be a lot of other species in there besides the one that we might eventually want to harvest, we need to embrace that.
Yeah, of course. Absolutely. Right.
Even the broadleaves, other than nitrogen fixers, they have some pretty profound influences on the soil, because most of them bring up a lot more base cations. And so they can, like calcium and magnesium. So they can really enrich that ecosystem. And if you start changing the chemistry of the forest floor and the soil by having those species in there, then Sue can explain how this affects the microbes. I mean, if you change the pH of the soil, if you increase the pH of the soil, you're going to get probably more diverse microflora as well.
Oh, really?
So
okay.
I think by excluding broadleaves, we are also simplifying and the below ground organisms and making them less diverse.
Yeah, and thinking about our one of our main species here on the BC coast, western red cedar, an important tree species. It's unlike most other conifers. It has a arbuscular mycorrhizal association. So these are the mycorrhizae that live within the root system.
Oh, okay.
And so these mycorrhizae are actually obligate. So they need the tree in order to survive, unlike the ectomycorrhizae that associate with Douglas fir, with western hemlock.
Right.
So these fungi can't survive without their host. And one problem that's been seen with a lot of the clear cutting that was done sort of 80 years ago in places like Haida Gwaii, where I've done research, is that there's been no host plants on that site since all the cedars were cut. And so now they have problems regenerating cedar. When they plant cedar on the sites, they're not surviving. And it's potentially because there aren't the inoculum of the arbuscular mycorrhizae. And a lot of our fern species, so sword fern, deer fern, they also have arbuscular mycorrhizae, but we don't know if they're the same species as we would find on cedar. So it shows the importance. Some of these these species, as I say, cannot survive without the living tree species. So
this is so important in how we manage forests, because we are so used to thinking about every species other than our crop tree species as a competitor, and not thinking about these other species that we would therefore brush out or herbicide or whatever. What effect are they having, especially below ground?
Sure.
And what is their effect on the whole below ground ecosystem? They're probably raising the pH. They're influencing the soil microbiome, which influences and directly influencing the soil fauna as well. So we need to think about more than competition. And especially if you put that whole ecosystem together, and not just what you see about how fast trees put on stems and leaves, but see that whole ecosystem. And imagine if I get rid of the aspen, let's say, if I get rid of the aspen, what effect is that going to have? And we can tell you this is going to reduce calcium, it's going to reduce magnesium, it's going to change it's going to change the microbes in the forest floor, it's going to change the fauna, it's going to change that whole ecosystem below ground. So we need to think more carefully about these as parts of ecosystems and not just competitors for nutrients or water or light. Yeah,
absolutely. Yeah, no, that totally makes sense. The ecosystems that are unseen are equally as important, right? And as Sue mentioned, the example, even the economic example of places where, I think you said 80 years ago, or we'll say decades ago, whatever, there was clear cut harvesting of cedar, and now it's not coming back because it doesn't have the correct microbiome to support it again. And like that's a direct economic problem there for folks that are economically inclined to think about things those ways, right? So there's an argument from all sides of the spectrum there, and I think
especially
it's important
if you think long term. We have to think, yes, our next crop will grow faster, get free to grow faster, and may reach economic rotation that much sooner if we brush out these other species. But what are the long-term implications for the productivity of that site and the fertility of that soil, which is largely created by all the organisms in it? So what's the implication 100 years from now of you taking out all the other species? Yeah,
absolutely. And that's what true sustainability is all about, right? We're talking about sustainability of all the values, and this is in perpetuity, forever, not just for the foreseeable 100, 200, 300 years, but beyond that. And
to imagine we could actually improve that site.
That's the other thing.
We're so busy trying, we're not going to degrade it too much. And if we mimic nature, then we won't degrade it any more than, nature would. But we could actually improve that site if we thought more about how we're managing it.
We could
improve the fertility of the soil, the diversity, the resilience, all by just thinking about that ecosystem and what good we could do for that ecosystem.
Yeah. I liked, there was something in your talk, Cindy, about previously we only thought about soils as don't lose it and don't squish it. Right? We don't want to compress it. Yeah, exactly. We don't want to compress it, and we don't want it to erode. So we don't want to lose it, we don't want to squish it.
That's pretty much our regulations for forestry when it comes to soil.
It is, for sure. And
don't lose it, don't squish it, throw a log on top, walk away.
Call it a day. And I think in some sense there was probably some sense in that, right? There was a lot of ideas. The fact that we were thinking about that back then is great. We've moved on now. Now we know that there's so much more to this, and policies need to shift to encompass these new understandings, this new science, right?
It's a living ecosystem down there.
Yeah. No, absolutely.
It's not just the stuff that holds up the trees.
Yeah. Yeah, exactly. Yeah, it holds the water long enough to let them grow. Yeah. Yeah, it's absolutely crucial.
It's not dirt.
Yeah. Yeah, what was it? My soil prof in school, he said dirt is the thing you find on your kitchen floor. Soil is what's outside. I'm like, yeah, that checks out. I like that. So I use that now. I even got my 4-year-old saying stuff like that.
Excellent.
Learn early, right? Gotta indoctrinate them into the ideas. Yeah.
But we should be thinking about this every time we see just the complete lack of care with which we get in some big yellow machine, whether it's in a forest or, when we're building
the mines.
Or, just anywhere you go where they're building a house or whatever, they just scrape it away like it's just some stuff that's in the way, and then, plop something on top and pound it down and think, do you not know what that was? Do you not know what it's got layers, and the top of it is a whole ecosystem, and you're sticking that down there and just throwing whatever on top and pounding it down. It's like, this is not dirt. This is not just a physical substrate that we can just move around and to our pleasure. This is an ecosystem you're messing with here. Yeah. It will make you insane. Actually,
I can feel the frustration. Yeah,
the complete lack of care with which we treat soil. It's just shocking once you know about it.
Yeah. Absolutely. No, that totally makes sense. I recently saw there was this beautiful fen that I think it happened to have some pitcher plants and stuff close to the city here in Edmonton. And one day it got bulldozed for a ball diamond. I was like, oh no, you guys, what are we doing?
You weep, and the more you understand about it, the more you weep for the way it's treated.
Yeah, it's
all those organisms,
pretty inconsiderate.
That whole ecosystem, that was an ecosystem. Oh, let's put a ball diamond here.
Yeah. I want to throw a ball. Yeah, it's absolutely wild. So I think I want to finish off by getting into this concept of. We've been talking about it the whole time, but someone put a great name to this: the continuous root forestry idea. Right? I'm assuming that's kind of what we've been talking about, this idea of having continuous living root systems across harvested areas to maintain a healthy, active, and living soil microbiome that is contributing to, the above ground ecosystem as well. Right? How do we describe this idea of continuous root forestry, and how is that connected to regenerative forest practices?
Yeah, it grew out of recognition of the benefits of continuous cover forestry, which is being done now in many places, and which is sort of a variant of retention forestry. It basically just means leaving some of the trees, but it's called continuous cover because we are so focused on what we see above ground, right?
Yeah, trees.
And that the only thing that matters is light. And this is such a frustration when you know that trees aren't usually limited by light; they're limited by nutrients or water, and those come from the soil. So we shouldn't be thinking about
Love that thought.
The above ground portion all the time. So once we started realizing this stuff about the importance of living roots, I thought, this is the message we've got to get across. There's that above ground half of the ecosystem, and there's this one too. And I think a lot of the benefits of retention forestry or continuous cover forestry are because they're living roots in the soil. So yes, there's cover, and that provides lots of nice stuff and has huge biodiversity implications. But below ground is at least as important. So that's the message I thought we should get out with calling it continuous root forestry, just to force people to think, the other half.
Right. The hidden half. Love that idea of the idea that we've simplified it to the availability of sunlight, whereas the nutrients and water availability are more, or at least I'd say more important, or at least equally as important as sunlight availability, right? And we just completely ignore those.
If you're a forester or whoever you are, just visualize. You got that above ground, and as the forest is developing, you can see that, but imagine the other half. And as the canopy, the crowns are expanding, the canopy is closing, it's going on below ground too.
and that's affecting the availability of water and nutrients, and that's what's making that tree grow up. It's not because it wants to reach the sun.
Yeah.
It's going for the light. No, it's not. It's getting enough water and nutrients because of what's happening below ground with all the organisms and its root system, its mycorrhizal fungi. That's providing the resources it needs to grow up.
Yeah.
Not because it's trying to get to the light.
Oh, race. Yeah. Gotcha. Interesting. I love that. So I would like to kind of summarize everything we've talked, and I found that you guys had made a pretty great summary of recommendations for how to consider, how to think about, and what to do as far as altering forest harvest strategies and that kind of thing in regards to thinking about continuous root forestry, keeping those root systems healthy. So I think, like, number one, the concept of just living roots, the 10 meter thing, I think that's so achievable and accomplishable. It's just like, I think it's already being done in a lot of places, maybe not 10 meters, but there's a certain amount of continuous cover, right? Like, I think, yeah, why not? Absolutely.
It's not being done.
Oh, okay. Yeah.
In any places at all.
No. Okay.
Even Sweden, where we thought they were doing well, it turns out they're leaving like 5 trees per hectare or something.
Oh.
If you're not leaving about 30% of the stems, then you're really, you've got too much of that area that's depauperate in the flux from the trees. So what we see in British Columbia, if you look, from Google Maps, whatever, what you can see is often a clear cut with a patch of trees in the corner, and that's not what we meant by retention forestry. It means a significant amount of that cutover needs to have the influence of living trees. And we used to think it was a tree height, whatever, which is, again, thinking about light only. We now know it's not a tree height. It's 10 meters. Everything beyond 10 meters is out of the influence of living trees and is essentially what we would call a clear cut. So just minimizing the proportion of the cutovers that is more than 10 meters from a living tree. Just thinking about that when you're planning the operation. Think, okay, I could sustain this other part of the below ground ecosystem if I just leave enough trees that there's not much that's more than 10 meters from a tree stem.
Gotcha. I like that 30% idea too. That paints another picture, giving two ways to measure it, just to think about it, conceptualize it. 30%.
Yeah, that's based on the evidence that's in the existing literature. Not a lot of studies, unfortunately, but if you look at all the studies globally, it looks like that's the minimum to really retain the below ground diversity and functioning. Beyond that, you've changed the system a lot.
Gotcha. Okay. That's good. Perfect. And then number 2, you had fertilizer. We haven't talked about fertilizing besides using, nitrogen fixing shrubs and that kind of thing. But my understanding is that the concept of fertilizing, you had said even adding it, additive. Why that? Why is that important? Why do you think that's?
Yeah. The interesting thing about adding nitrogen to a forest where the trees are kind of moderately nitrogen deficient, like most of ours, is that it really increases above ground growth, which is great. But what that does is really reduce the flux of carbon to the roots, because if the trees have the nitrogen to produce more leaf biomass, they're going to do that. And there's only so much to go around, so they don't have any surplus photosynthate left. It's all being used there. And what we see is a huge decline, like a 60% reduction in one study, in the amount of carbon going below ground. And that's what results in the reduction of the mycorrhizal fungi. So yes, you've given the tree the nitrogen to make it grow bigger above ground, but you've reduced that below ground production
and that below ground, you change that below ground ecosystem. And then we see as the nitrogen is used and its availability declines, then you get that flux back. So, and, I've done quite a bit of work with nitrogen fertilization, and it is a great way to make trees put on above ground biomass, leaves and stem more.
Yep. Right.
But if we're doing that in these days of droughts, especially, we need to think about if we really want to compromise the below ground system, the fine root production, the mycorrhizal production, do we really want to compromise its ability to access water and other nutrients by forcing it to produce more above ground biomass? So in some cases we might.
Gotcha. So it's a balance. You don't want to fertilize everything. There's certain situations where if you don't have those nitrogen fixing plants, you might want to add some, but it's not. Yeah.
So
more is not better.
It suggests to me that maybe we should at least think about this and think about whether it would be better to use nitrogen fixing plants rather than this big sudden dose of fertilizer, which makes the tree adjust all of these things, and then has quite profound implications for the below ground ecosystem until everything gets readjusted. So do we want to shock that ecosystem like that, or do we just want to promote the accumulation of nitrogen, including nitrogen in soil organic matter, by having nitrogen fixing plants in there?
That makes sense. Okay. Well, could you give an example of like a situation? Is there any place in where you would want to do like add fertilizer, or is it just you're focusing more on actually on the nitrogen fixing plants in particular? I think that's a longer term, better, healthier idea.
Yeah, but in severely deficient situations, what adding the limiting nutrient can do is push that plant up to the point where it is photosynthesizing enough that it can put on more biomass, and it can also produce surplus carbon. So we saw this in some really nitrogen and phosphorus deficient sites on northern Vancouver Island. If we add nitrogen and phosphorus to those, we get an increase in tree growth, and we also get an increase in the diversity and abundance of mycorrhizal fungi, which is kind of weird because that's the opposite of the decline that you usually get and the change in mycorrhizal fungi. So I think in that case where it's that deficient, you can promote both above ground growth and below ground diversity and function by adding the limiting nutrients.
Yeah, there's a pocket of nutrient availability you want to stay in. You don't have too little, too much, both bad. So gotcha. Okay.
But sites that are that poor, maybe we don't want to use forestry. It's gonna cost a lot to get them functioning.
Fair enough.
So
absolutely.
We just need to think more carefully about the entire ecosystem when we're adding fertilizers.
Makes sense. Okay. The third point, and cut in if I'm getting these points wrong or if you've changed them since I've read the documents. You got diverse tree species. Well, we've explained that a lot. The importance of each tree species has its own specific footprint of microorganisms related to it. And having a diversity of those allows for more biodiversity and more hedging our bets and all of that, just being certain that we have options and we have a resilient forest soils. Yeah. Okay. And then also diversity of forest types was your fourth one. So by forest types, I'm assuming you meant, because we already talked about species, you were talking more about the stage, the age class of the forest. So,
So you can have diversity either at the stand level or at the landscape level.
Of course.
Preferably both.
Right.
So this is diversity at the landscape level.
Yeah.
Because then if you have that diversity in tree species and plant species, then you also have that diversity below ground. And so as systems are getting older, as the trees are aging, then you have that inoculum in the environment that it will be able to mature into the same sort of system that the existing mature ecosystems
are at. If that, and we've seen that with the mine site studies, that the farther that inoculum is away, the longer it takes for that ecosystem to develop. So just landscape level planning for diversity, and that's a good reason for so many other reasons.
Well, I would even think at a stand level even too, right? So if you're planning a forest harvest, you could leave, like you said, you're leaving continuous root forestry, right? So we're leaving a certain amount of mature trees in the landscape, we'll call it 30%. But you could also leave, if there's advanced regen underneath, you could leave some of that. And then you're going to go in and plant. That's going to give you more. I'm assuming that those different age classes, even within a stand, probably have their own footprint.
Very true.
Is that okay? Yeah, perfect. Great.
Value that advanced regen.
Yeah. Interesting. Okay, good.
Even
and then
if
the
it's
final,
not your crop tree.
Yeah. They're all important, right? Yeah, like you said, again, diversity of species, even shrubs and forbs and all that kind of stuff.
They're all part of the ecosystem, and if you get rid of them, then you probably get some invasives, and then you've got a bigger challenge.
Yeah,
so
totally.
Think about all those species that are there and manage them all.
And the final point you had, the fifth one, was monitor soil quality. So let's get into that. What does that mean? What does that look like? How does one do that?
Yeah, that's kind of a pushback to this idea that if we don't see a decline in stand productivity with successive rotations, that the soil must be fine. But built into that maintenance of stand productivity is using improved genetic stocks that's programmed to grow faster, as well as silvicultural techniques.
Right.
So we don't actually know if the soil beneath our successive rotations is degrading.
Yeah. That's not good.
Yeah. So I think if we're going to ask that question, is the soil at least being maintained? We need to actually monitor the soil.
Gotcha. How do you do that?
Well, I would suggest we need to monitor soil biodiversity, which is tricky. You can also measure rates of processes, and that's tricky too because, is a higher rate of decomposition better or worse?
Right.
We don't know. Is staying the same is probably the best. The amount of soil organic matter, and not just litter and slash, but the amount of mineral-associated organic matter, I thought is probably the most critical.
I'd agree. Yeah.
Okay.
Must be true then.
Well, there we go. It's a consensus, people. Yeah.
So at this point, when that is at the state of what we're able to monitor and what we can understand about it from what we measure, I would say let's think about that, because if you're just measuring carbon, you're going to get a whole lot of carbon after you clear cut because you've got all those dead roots, you got all that slash on top. There's a lot of carbon there. It's all 50% carbon. So yeah, you got lots of carbon, so everything's fine. No, it's not. It's not okay. It's that persistent pool in the mineral-associated organic matter that's being degraded by the microbes or processed by the microbes. I think that is probably the most critical one to monitor, because I think if you're maintaining that through its being regenerated, at least at the rate at which it's degrading, then that soil is probably okay.
It's balanced.
Yeah. And the DNA barcoding, this gives us the ability to get a really amazing picture of the level of biodiversity in the soil. But again, I don't know if what level of change in that whole community, massive community, is good or bad. So I don't think we're able to use that yet.
Gotcha. Okay.
No, but maybe targeting some of we've done in the past is looking at functional genes, abundance of functional genes. So ones that are important in what we call some of the narrow processes that are important, nitrification and lignin decomposition, carbon cycling. So focusing on some of the key processes, and then you can, using quantitative PCR, look at abundance of, like, how abundant those genes remain.
Right.
And that relates directly to the functionality of the ecosystem. So yeah, that's a good one.
Gotcha. Well, I think I want to summarize everything just to get everything out there and kind of just like get a good feel for the conversation we had. So ultimately, continuous root forestry is the goal. So we want living root systems across forest harvested areas to help promote living and healthy forest microbiomes. Adding in nitrogen-fixing plants when needed to, again, help promote healthy forest microbiomes, diverse tree species, diverse shrub species, and the diversity of stand types, so stand ages. And then we want to keep an eye on things and adjust course as we go. So I think those are all accomplishable, understandable, relatable, and good takeaway points. I always feel good about it when people can walk away from a conversation like this one with actionable material, right? Because then it's not just a forum. It's actually, this is something I can get advice from and something I can take away and maybe look into more and actually have conversations about in professional lives and that kind of thing. So,
and all of those will help us to develop or help resilient forests to develop. And that has to be the goal now.
Mm-hmm. Absolutely. No, you've converted me. I'm convinced. Let's do it. I'm in. I'm sure some of my listeners feel the same way.
Now we get the politicians to listen.
Yeah, that's right. Yeah. That's a harder, I don't know how hard can you twist someone's arm? I don't know.
Well, hopefully the policymakers, and I think that's the first step, is just to get the forestry policies updated and made fit for the 21st century.
Yeah, well, convince the professionals in that this is the way to go, and the forest policy comes next, right? Like that follows suit. So, conversations like these are exactly what we need, I think, to get this information out here. And I'm so thankful to both of you for taking the time to tell your story, to tell this story. This is novel to this podcast. I haven't discussed this at all. So I really appreciate your time and the clarity in which we can describe. Besides there was that one little sentence you said that I had completely lost me about radioactive X-ray photogrammetry, whatever. You lost me there, but that was only a second.
Lost me too.
Or whatever it was. But this was fantastic. And you both did such a great job of explaining your point of view and the science that you spent your lifetimes putting out there. So thank you so much for everything. I really appreciate it. This has been great.
Thanks, Matt.
Thanks, Matt.
Thank you so much for listening. I hope you enjoyed that. I hope you learned a lot. I know I learned a pile, and I'm going to have to dig into this deeper to really understand the consequences of our actions, the impact of what we're doing, and how we can move forward, what we should really be thinking about, how we can think about this differently. So, yeah, what a fantastic dive into the world of soil microbiomes, the life beneath our feet, right? So, if you have any questions for myself, for Sue, for Cindy, shoot me an email, yourforestpodcast@gmail.com, and I will get back to you. So, thanks a lot for listening. Catch you next time. Take it easy.